Hi, my name is Mary Lou Bailey, and I'm working in the lab of Dr. Mark Sherwin and my mentor Devin Edwards in the UCSB Physics Department, and I am a EUREKA intern, and I made sample holders for ultrafast electron spinners. So ESR investigates the unpaired electron spins in a solid or liquid in order to learn more about the structure or local environment of that solid or liquid that we're looking at. So for example, we can look at proteins and we can tell how far different places are on that structure. A prominent example is proteorhodopsin in biology. This protein forms groups of proteins, but we don't know how it does that. So by being able to tell how far apart electrons are or where certain things are on the hexamer, we can learn more about that protein or whatever biological molecule we're studying. So a little bit about the physics behind this. You have your electron spins that are either up or down in the sample, and that is in the magnetic field B. So let's say you have one that's spin up. You take a very high-powered source and excite the spin, and that causes the spin to tip and precess. And we're interested in measuring the amount of time it takes for the precession to come back up or decay. So the time it takes for the spin to come back up. And that can tell us how far apart the electrons are in a biological molecule. For example, if you have two electrons that are close together, then each of their magnetic fields affects one of the other. So the spins will come back up quicker, whereas if they're further apart, the spins will take longer to come back up. So ESR at UCSB is unique because we have a free electron laser which provides power at 4 times the magnitude more than what a typical ESR experiment would be powered by, and that gives us frequencies at over 100 gigahertz. So to match that, we have the magnet, which is 200,000 times the magnetic field of the Earth. And so all this high power and high frequencies and magnetic fields are important because it allows us to measure really fast decaying spins in a sample. So this is the UCSB FEL, and we have our electrons, and this is the accelerator that comes through here, and they're excited by the array of magnets, and that causes them to emit light, and then that's essentially piped into our lab where it's sent down the waveguide, which is how we study biological samples. So we have a sample holder which is Teflon, and it is cylindrical, and it was my job to design a more efficient one. So we put the sample at the end of the waveguide, which goes into the magnet right here, and then we send our FEL pulse in, and then we measure the decaying signal with the detector in that. It's out there. So our main problem is that the sample holder is reflecting FEL pulse back to the detector, which messes with our data of the decaying spins. So as you can see here, we have to wait 80 nanoseconds to turn the detector on, which is a long time for us to not be looking at data. So we want to be able to decrease this dead time. So to create new, more efficient sample holders, I do that in 2 steps. I first design a sketch in a CAD 2D program, which in this case is SolidWorks, and my first hypothesis is a double cone, because we think instead of having just the FEL pulse come straight down and straight back up, the cones will deflect some of that pulse. So I have the Teflon rod, and then we're trying a new material too, which is Rexlite, and it's another type of plastic. And I take that rod and put it in the lathe and machine in my hand in the machine shop. So here are some of the sample holders. These were the original ones in Teflon, and then Rexlite, which is the new material we were also testing. And then here's the Teflon double cone, the Rexlite double cone. As you can see, they're very small. The cylindrical ones are 5 millimeters by 5 millimeters, and then the double cone are about 5 millimeters wide by 8 millimeters long. So in order to test these sample holders, we put them at the end of the waveguide, empty though, and then we send our FEL pulse down. So our raw data is just amplitude versus time. So you can see this is the FEL pulse, and then this is when we turn the detector on, and then this is FEL reflection. But it's hard to kind of get an idea of what's reflecting what with the time domain. So we Fourier transform this, which just means we go from the time domain to the frequency domain, so we can see what frequency is being reflected, mainly the FEL frequency. So here is the cylindrical Teflon and Rexolite holders. You can see the FEL frequency is 500 megahertz. That's mixed down from 200 gigahertz, so the detector can actually see the oscillations. And you can see that the Teflon has a lot more reflection and a higher peak, and it looks like a higher area under the peak too than the Rexolite. So this suggests that Rexolite's a better material. But testing with a double cone, they look pretty similar from both Teflon and Rexolite. They look the peaks looks kind of smaller than the cylindrical, but the thing is we see two prominent peaks, and so we're not sure what that means exactly, so we can't necessarily conclude that a double cone is actually better. It might actually be worse. So to get a more general idea of what's reflecting more and what isn't, we can integrate the peaks and look at the area under to see how much is being reflected. So as you can see, the Teflon in cylindrical has the highest amount of reflections, and the cylindrical Rexolite is the lowest. But another thing to look at is that the Teflon counterparts of each design has a higher reflection than the Rexolite. So we can conclude that Rexolite is a better material. We're still, again, not sure if the double cone is better, but we'll probably switch over to Rexolite cylindrical holders. And then for future testing, we can actually add sample to these sample holders and then see if we can actually decrease the dead time when we measure spin decay. And I'd like to thank my mentor, Devin, and my PI, Dr. Mark Sherwin, Tyler, EUREKA, and the EUREKA program for providing me with the opportunity and funding to do my research this summer.